everybody, this is a presentation on habitat suitability models. So when we're modeling habitat suitability, we're really looking for what's the suitable habitat for a species. This is also called species distribution models and ecological niche modeling. The species on the left is one that I worked with quite a bit in my dissertation, an invasive species from North Africa and Central Asia. It likes to be in desert riparian areas, so it has a specific habitat where it lives, and that's where folks have been working to eradicate it in the United States because it sucks up a lot of water and changes the system. So habitat suitability models emerges from the idea that each species has a specific habitat that it can live in. In other words, it has environmental requirements that are only met by certain habitats and not met by others. Good example, this is here in northwestern California. We've got redwood trees, a very unique species that likes it wet, likes it cool, and can actually absorb water from fog, so that's one of the reasons it's here. At the other extreme would be something like a saguaro cactus from the US Southwest that likes it really dry or survives where it's really dry. So two very different species with very different habitat requirements or very different environmental requirements. So to predict habitat suitability, which I should say is important in a number of areas, if we want to preserve a species or if you want to monitor invasive species, I'll talk a little bit more about that in a minute. But that's why we want to do prediction. Typically we do this at large spatial and temporal extents. One of the reasons for that is it's easier to do it over large extents. It's harder to do it in small areas because we simply don't have the range of environmental variables, the range of characteristics to identify a species' unique habitat requirements. The problem is most of our data has unknown uncertainty, most of it is biased, and most of it is just occurrences. In other words, these large spatial data sets like we find in GBIF, usually it's just, oh, here's where we found something with very other information. No vigor, no height, none of the other variables that we've been using in the semester. There's also lots of different species, and we'd love to do this under climate change for a lot of species. So there's a lot of different methods. Some of these you can use if you have the data, like density and abundance. If you have a continuous response, you can use the other methods we've talked about. If you have presence-absence, you could use a GLM with a logit or logistic. But then you have to ask, what does an absence mean? In other words, does an absence mean the species can't live there, that that's not good habitat, or does that mean that it hasn't gotten there yet, or that someone cut it down, removed it? So usually what we end up with is presence only, and the problem here is, what do we regress against? In other words, they're just presences. It's like only having ones and no zeros. So that creates a challenge because we really need to have something to regress against. So initially folks were talking about something called pseudo absences or background points. Pseudo absences has kind of died out. I think that's a good thing because that's not really what they are. What we're really doing is we're regressing the points based on density of where they are against the entire area, or the entire area of interest. And Jeff Dunk, shout out to him because he called this a point process problem, and I think that fits well. In other words, it's like density, but it's density over an area. The theory for this is well documented in the Theory of Biogeography, foundational book from Brown and Limono, highly recommend it. And the basic idea is that species grow better, like here with salinity. Species are going to have a certain range where they do better with salinity. Similarly, they're going to have a range where they do better with temperature. Same thing with precipitation, other environmental variables. When we put these together, so these are our traditional response curves, and over here we put them into a two-dimensional chart, and say as salinity increases, as temperature increases, there's going to be a niche where our species does well. That's this green area. It's the right combination of environmental requirements for that species. And then as you move away from that niche, it's not going to do as well, and we move outside of that niche, the species is going to die. So that's the environmental niche for that species. There's a lot of different examples for this. I'm not going to go into these too much, but there's a lot of papers out there to check out. These become very powerful tools for predicting the potential habitat. And that's important to remember. This is the potential. It's not the realized. It's the potential, okay, based on changing conditions, including climate, pollution, water, and a lot of human impacts. But because they're based on environmental data, they don't predict the actual distribution of the species. They are called species distribution models, or SDMs, but it's not going to say where a species is or where it's distributed. It's going to say where there is habitat, so potential. That's important to remember. A fun paper on this was one on the distribution of Bigfoot. I recommend checking it out. It actually is a serious paper looking at the distribution of Bigfoot based on occurrence data, and also looks at brown bear occurrences and compares the two. You can draw your own conclusions from that. There are some real issues with HSMs. Human-modified environments like polar bears in Denver mess up the models, as do isolated areas where species may just not have reached there yet. Early attempts with this, BioClim is actually a modeling package as well as the data we've been using. Biomapper, GARP, GLMs, GAMs, different methods to modify occurrence data and use these methods with it, usually around density. The methods that are really being used now, by far the most dominant one is Maxent, with over 1,000 papers using Maxent to do habitat suitability modeling. Very, very popular. We'll use it next week. And then HEMI, or hyperenvelope modeling interface, the one that I created. We'll talk a little bit about that this week. NPMR not being used a lot, but it's another one that's interesting. You can use tree methods, but we get into some of the same issues that we do before with overfitting. Okay, so what I do want to talk about is there's two general approaches here. One on the right, called mechanistic, is a more traditional approach used a long time ago before we had Maxent, and then one based on occurrences. And this is where Maxent and HEMI come in. And the idea is you take occurrences from the field where you've gone out and found where a species is, recorded that location, correlate that with some environmental variables. That creates our model, just as we've done in the past, except our occurrences are simply ones and zeros. And then from that and predictor layers that represent our entire study area, we can generate a map, just like we've done before. The difference being the modeling package we use. On the other side are greenhouse experiments. In other words, if you don't have occurrence data, you take and in a greenhouse, you change the temperature and precipitation requirements for a species. You do that along a gradient. You see where the species does well and where it doesn't. From that, you can actually design a model that will then generate a map. Now, these each have trade-offs. If you have the occurrence data, in other words, you have widespread occurrence data for a species, the one on the left is going to perform better, but we rarely have that. So the one on the right, which are usually only used by the agricultural industry, seed producers, things like that, they could be important because they could provide you with a larger range of requirements than you're going to get from occurrences if you have incomplete occurrences. But they're also not done much within the natural resource science community. So as a couple examples, here's good old Doug fir, and we can see that Doug fir, when we look at it against temperature, there's a certain range of temperatures. It's not in the coldest areas up here in the high Sierras, okay, but it is in colder areas in the northwest, and definitely not down in the desert. All right, key to understanding this and how habitat suitability models works are these histograms, where in green I've histogrammed the distribution of temperature for all of California. So green represents histogram pixels, so 200, it looks like there's a lot around 160, remembering this is temperatures, mean temperature in degrees C times 10. So these are our deserts, where it gets pretty hot. And then in red is a histogram of the temperatures where Doug fir was found. Okay, so red is Doug fir, green is all of California. What we can see is that Doug fir is biased towards the colder areas, not the coldest down here, but definitely the colder areas of California. Both these histograms have been scaled to go from 0 to 1 so that it's easy to see. Otherwise the Doug fir one would sit down here underneath the green one and it'd be harder to see. So I've just scaled them up to go to the same height. And then the black line represents a response curve for a model that was created within HEMI2, which we'll talk about a little later. The key thing to look for here is the difference between the two. Because these histograms have a difference, that shows that Doug fir has a habitat preference for colder areas, and that there's areas where it appears to not do well, these higher temperature areas. Alright, if we look at precipitation, similar. Doug fir likes it wet, not down here in these dry areas, low precipitation. And here when we histogram that, we see an even more extreme effect where Doug fir does not like it dry. Doug fir does well where it's wetter, in fact does quite well. And again, the red being the Doug fir, histogram of the Doug fir occurrences, where the green is a histogram of all of California. And the black line is the response curve from HEMI2. Okay, so one of the important things about this and about the data that we have is the difference between the occurrences that we have, the realized niche and the fundamental niche. And you're going to want to know the terms realized and fundamental. So our occurrences, that's where you've gone out and you've said, oh, here's a Doug fir, here's a Doug fir, here's a Doug fir, recorded the locations for that with the GPS, brought those in as a data set, and those are represented by the little plus marks on these graphs. The realized niche is where all the Doug fir are. So it's where it's gotten to. And so the circle, the gray area in the chart in the upper left, represents where Doug fir actually is. Okay, and it's just a diagram, but you can imagine this as being 3 islands. The fundamental niche is the potential distribution. It's all the areas that Doug fir could live. So let's say these are 3 islands off the coast of California. It could be in all 3 of them. It's on this one, fairly over most of it, covering this one, but it's never reached this island. Now, we take these occurrences, we create a model, we then move into an environmental space like I showed before, where we've got 2 environmental predictors. These could be temperature and precipitation. And then we create our model, which is in blue. And this shows that our model will always be a subset of the realized niche, because we're never going to get occurrences for all the Doug fir. You might for some species that's dying out, but that still probably wouldn't represent its realized niche, because there's probably reasons that it's dying out. Unless it was near extinction for natural causes. Anyway, so our blue is our model, the gray again, the realized niche, the black line being the fundamental niche, where it's real, that's really the environmental space that it occupies or can occupy. And then we move that back into geographic space, and we see that we can model where our occurrences are. We can model some of the area where it could be, but not all of it. And in fact, we can model where it is, but not all of it, because we haven't collected data, and maybe want to collect some more data in here to fill out our model and make it better. We can also predict where it could be and it's not. So this is very important with invasives and endangered species. And I'm going to stop there and continue on another